Now that we've learned how the SVD works, we're going to use that to create an MDS plot, a multidimensional scale plot. So what I'm going to do is I'm going to apply it to a version, a subset of our matrix that has the tissue gene expression that we've been working with. I'm only going to look at kidney, colon, and liver just to keep it simple. And what I'm going to do first is I'm going to compute the singular value decomposition on this subset. Oh, right, so on this subset of the matrix that has only 99 columns, because again, it only uses these 3 tissues. Okay, so I want to make a plot. I want to be able to plot all these samples and see how close they are to each other. I want to see if kidneys are closer to kidneys and colons, all that stuff, okay? So I can't make a picture of 2 points that have 22,000 dimensions. I'm going to use singular value decomposition, and I'm going to look at the 2 dimensions with the most that have the most variability. The columns have the most variability. So what we saw in this video is that we can take the SVD of MAT. Now I'm going to remove the means. I'm going to detrend them in this sense because I don't want the differences in means to contribute to the variability that I'm capturing here. When you compute distance between 2 columns, the mean of the genes doesn't come in because they cancel out when you subtract them from each other. All right, so I'm going to take this SVD. Look at how fast this is. It's done. Okay, and now what I'm going to do is I'm going to reconstruct the Z matrix that has just 2 dimensions. And the way we did that is we saw this formula that gave us this way of doing it, right? So we take just the first 2 columns, and that is Z. Oh, I need to add the S here. And now we can see that this Z has dimensions 2 by 99. So instead of 22,215 dimensions, now I'm down to 2, and I'm ready to make pictures. Now I'm going to transform the Z just because I'm more used to plotting when I have things in columns, but you know there's no reason to do that. I'm just used to it doing that way. Okay, so I can plot. So for example, then you can do this. Right? So you can see all the points here, and now the idea is that if I want to know the distance between 2 columns of MAT, which is these are 22-dimensional vectors, so it's not easy to do. I instead, with this visualization, suppose the idea is that that distance is approximated by the distance between points in this plot. So now I can visualize it. That's the power of this approach. So let's look at this a little bit closer. So I'm actually gonna add this library to make it a little nicer looking. And now I can plot Z. All right, so you see the points, and now what do you think these 3 clusters are? Well, let's add some color to this and see. I'm going to add the tissue as color, and let's see what we have. Well, there you go. So we have that the tissues separate out as we expected. So that is that's a great feature. Now we don't have to restrict ourselves to the first 2 dimensions. For example, you can look at the 3rd and the 4th. Here's the 3rd and the 4th. Okay, and now we do that instead, and we see something different. Now we have another source of variability here, and you can see that one of these tissues, the orange tissue, is splitting up into 2. That could be sex, for example. It could be some other variable, but it also has quite a bit of variability, but not as much as tissue-to-tissue variability. That was the strongest one. It seemed to capture the first 2 columns of the singular value decomposition. Okay, now one other quick thing is that we don't have to do this with the singular value decomposition. There's a function called the CMD in R. It's the multi-dimensional scaling plot function that gets you that. So if I compute the distance between things, then it uses CMD scale to approximate the distances, and then we get practically the same picture. Well, I don't want this to be so big. Let's get rid of that. But yeah, you get this. We get the same plot. It's doing practically the same thing. Another little point is that you can change the K. K2 is the default, so it only gives you 2 dimensions, but you can change this to 4 or 5 or 6 and get as many dimensions as you want from the SVD approach, the columns of U and V. Okay, so that's how multidimensional scaling is done in R and how it relates to the singular valley decomposition.